If someone were to mention foodborne illness, two thoughts might have crossed your mind. Either salmonella on raw chicken, or E. coli recalls that have been in the news recently. The latter will be our focus for this video. Shiga toxin, found in some pathogenic strains of E. coli, is a toxin which can hide in the gut of various species of cattle, deer, swine, and even infected humans. While it is non-toxic to the host animal, it is incredibly toxic to humans, and is one of the most potent bacterial toxins known. While some critical aspects of the pathogenesis of the toxin are still a mystery to us, it is known to cause abdominal pain, diarrhea, and in severe cases, hemorrhagic colitis or kidney damage from hemolytic uremic syndrome. In order to hone in on various strains of STEC, we use serotyping, since it's able to identify strains of bacteria from unique flags on the cell's surface. These serotypes are determined by two kinds of antigens on the cell's surface. The small, almost fuzzy coating on the entirety of the cell is known as the O antigen. The long, strand-like structures on the cell's surface are flagella and are called the H antigen. The numbers after the O or H refer to combinations of antigens in each serotype. These antigens can be analyzed to help detect the presence of the pathogen through the use of PCR in the field. By now, I bet you're curious about how in the world E. coli outbreaks can be identified and treated. Let's take a look at that now. If an outbreak is detected, our major task force for such circumstances, the CDC, works tirelessly to determine the source, as well as what can be done to prevent it from spreading further. Today, we will be examining an outbreak of E. coli O157H7 on romaine lettuce grown in the Salinas Valley of California from November of 2019 to January of 2020. In total, 27 states and 167 people were affected. Surprisingly, no deaths were reported. In this outbreak, the CDC employed epidemiological laboratories and traceback methods to identify the outbreak. When the epidemiological investigation begins, the CDC notes trends and patterns in geographical distribution and establishes a theoretical start and end date for the outbreak consistent with noted cases and timeframes from past outbreaks from E. coli O157H7. Simultaneously, traceback investigations also begin, and potential sites for contamination are investigated. Those who contracted the illness are interviewed about shopping habits, restaurants visited, and more to pinpoint potential sources for the outbreak. Once an intermediate source, such as a store or restaurant, has been identified, the CDC works to identify the primary source. For this outbreak, the CDC traced the pathogen back to a farm in the Salinas Valley of California. In the laboratory, the CDC utilizes PulseNet to DNA fingerprint the bacteria using whole genome sequencing, or WGS, to look for cases with genetically similar bacteria. Like several other foodborne illnesses, E. coli has a habit of targeting the weak. The sickness it causes from the Shiga toxin is most dangerous to immunosuppressed individuals, the elderly, and young children. Typically, if there are deaths from a STEC infection, it is from one of these demographics. It works quickly. Symptoms will usually begin after 3 to 4 days and last for 5 to 7 days. However, it is also bad at keeping hidden. Healthcare providers can easily identify it due to the persistent vomiting, bloody diarrhea, and fever greater than 102 degrees Fahrenheit, or 39 degrees Celsius. After the symptoms have been identified and a STEC infection is suspected, the lab work begins. In stool samples, STEC can be identified through traditional culture-based methods by inoculating samples on sorbitol McConkey agar, which is both selective and differential. The Shiga toxin E. coli cannot ferment the sorbitol sugars in the agar, and other strains of E. coli can ferment it. This result shows the generic E. coli colonies colored bright pink, while the STEC colonies are colorless and gray. Companies, farms, food processors, and governmental organizations like the USDA are all working to learn more about STEC, both from a microbiological and medical perspective, and how it relates to the food industry. The research is continuous. However, for consumers, the most important thing to do to avoid a STEC infection is to follow USDA recommended cooking temperatures and times for the more susceptible products, and get help if you think you're infected. And there you have it. And that's how we hone in and eventually eliminate E. coli infections and outbreaks.